My name is Lauren Testa, and I'm here because I'm a scientist in the rare disease field. I'm a fourth year PhD candidate in cell and molecular biology at Penn. I'm also a rare disease patient. I myself have Marfan syndrome, one of the two genetic connective tissue disorders that I'm trying to develop therapies for during my PhD. Patients with Marfan syndrome experience lifelong, often progressive symptoms within the skeletal system, eyes, cardiac valves, and aorta. All of these issues can reduce quality of life, and the aortic complications of Marfan syndrome, aortic aneurysm and dissection, can be life-threatening if not properly monitored and surgically repaired. Being bounced around between cardiologists, orthopedists, podiatrists, ophthalmologists, and geneticists throughout my life deepened my interest in the medical field and led me to want to be a doctor to help patients with genetic conditions. There was only one problem. Like the vast majority of genetic disorders, Marfan syndrome is, at least currently, incurable. So I decided that if therapies for rare diseases didn't exist, that I would have to be the one to make them. After college, I worked as a postdoc research fellow for 2 years in the Undiagnosed Diseases program at the NIH Clinical Center. I then enrolled in a PhD program at Penn. One day during my first year, I was in a meeting with Dr. Kiran Musunuru, who taught my gene therapy seminar class. Kiran has spent his career developing genetic therapies for both rare and common diseases. I expressed my enthusiasm about his work and disclosed my Marfan diagnosis. He offered for me to work in his lab on trying to correct Marfan-causing variants, including my own, so of course I said yes. What does it take to make a successful genetic therapy for rare diseases? At its core, there are two steps. Step one is coming up with a strategy to precisely and efficiently fix a harmful variant in a patient's DNA. Step two is delivering that strategy to the relevant cells within the patient's body. Let's focus on step one for right now, which involves using CRISPR to correct DNA misspellings or variants that cause genetic diseases. There are two components to CRISPR, a protein shown here in white, red, and salmon, and an RNA molecule, a so-called guide RNA, shown here in purple. The guide RNA acts as a GPS that tells the protein where along the 3 billion letter long sequence to target. The RNA and protein come together and scan along the DNA, shown here as a blue and green double helix. When the CRISPR machinery finds the disease-causing variant specified by that GPS, it stops there. The red and salmon portions of the protein convert the harmful variant into the correct letter. The beauty of the system is in its reprogrammability. By tweaking the guide RNA, the GPS, you can retarget the CRISPR machinery to virtually any other spot in the genome. CRISPR could theoretically be used not only to correct my own Marfan variant, but the thousands of other Marfan variants and millions of other variants that cause genetic diseases. I've made some progress on Marfan syndrome throughout my PhD, but I've run into the same issue that faces therapeutic development for so many other rare conditions, which leads me to step two of the genetic therapy development process, delivering the CRISPR components to the cells in the body that need to be fixed. No matter how good CRISPR machinery is at rewriting DNA, we currently don't have great strategies to deliver that machinery to many disease-relevant organ systems. To highlight this, let's talk about where the most progress has been made in the therapeutic gene editing space. The main organs that we can readily target with our current delivery methods are the eye by direct injection, the blood by collecting blood, rewriting the DNA, and returning the blood back into the patient, and the liver by administration into the circulation. One of the jobs of the liver is filtering substances out of the blood. So CRISPR therapies that are administered intravenously often get taken up by cells in the liver. This is all good news for patients with rare diseases affecting the eye, liver, and blood, and I fully believe that victories for these rare diseases help the whole rare disease community. At the same time, I think that prioritizing therapeutic development for only the the diseases that affect these tissues can fuel inequities within the rare disease therapeutics field. What about patients like me with Marfan syndrome and other multisystemic connective tissue disorders who would need the therapy to access the skeletal system, aorta, heart valves, and so much more in order to feel better? What about patients with neurological disease whose therapy would have to penetrate the blood-brain barrier to be effective? I think there are several solutions to this problem. The first is to wait for the development of new delivery methods that can target the heart, vasculature, brain, lungs, muscles, and so forth. I can assure you that there are many great scientists working on this, and we've made a lot of progress towards increasing the types of tissues that CRISPR can access. But what if, with a bit of creativity, we can improve the lives of those with multisystemic diseases using the technologies that we've already tested and validated? Take the example of transthyretin amyloidosis, a rare disease caused by toxic protein buildup in the heart and nerves. While patients experience cardiac and neurological symptoms, the toxic protein that builds up is actually made in the liver. This means that targeting a CRISPR therapy to the liver alone should be able to restore healthy function of the liver protein, which would prevent its toxic buildup in the heart and nerves and ultimately improve symptoms of the disease. As a matter of fact, Intellia Therapeutics has already done this and has shown a 90% reduction of this liver protein in the blood of patients with transthyretin amyloidosis who received a one-time treatment of this therapy. Now let's apply the same concept of liver-directed gene editing to connective tissue disorders. While the gene that causes Marfan syndrome is turned on throughout the entire body, there is another multisystemic connective tissue disorder called pseudoxanthoma elasticum, or PXE, caused by variants in a gene that is only turned on in the liver. This means that, again, targeting CRISPR to the liver alone may be enough to improve the multisystemic symptoms of the disease, just like transthyretin amyloidosis. Around a year and a half ago, Kieran and I, along with Dr. Lily Lee at Thomas Jefferson University, started a new project aimed at answering this question. My work on PXE is tangentially related to our lab's work in the Somatic Cell Genome Editing Consortium, an NIH-led and funded initiative of researchers across the country who aim to bring gene editing therapies for rare diseases towards the clinic. PXE shares some similarities with Marfan syndrome, but rather than the connective tissue becoming fragile, it develops excessive calcification. Patients with PXE experience progressive vision loss due to retinal calcification, as you can see by comparing a photo of my very own healthy retina with the retina of a PXE patient. Likewise, arterial calcification can lead to blockages within arteries throughout the body, which can be life-threatening. In healthy people, a protein in the liver prevents connective tissue from calcifying. Patients with PXE lack this liver protein, so they experience excessive calcification of connective tissue within the skin, arteries, and eyes. I don't have time to talk about the details of my work today, though I'd love to chat offline with anyone who wants to hear more specifics about the science. The main takeaway I want to leave you with is this. We've made a mouse model containing a real-life human PXE variant and treated them with a CRISPR editor protein and a guide RNA, the same guide RNA that would be administered to patients carrying this variant. We've shown that this therapy was able to efficiently correct the disease-causing variant in the liver. We then looked at connective tissue calcification within the muscles of these mice. Since, like PXE patients, the skin is the first organ to calcify in our mouse model. To measure muscle calcification, we performed CT scans of the muscles of healthy mice, PXE mice that received no treatment, and PXE mice that received a one-time dose of the CRISPR therapy. Healthy mice show no calcification as we expected. Untreated PXE mice show lots of calcification, which you can see by the white squiggles in the image. The calcification has been almost entirely prevented in our treated mice. In other words, this is the first example of a one-and-done liver-directed gene editing therapy to address the multisystemic manifestations of a connective tissue disorder. As excited as I am by this progress, and trust me, I absolutely lost my mind when I saw this result the first time, being a patient scientist has been a complicated journey. As someone living with a genetic connective tissue disorder myself, I can't help but to think of the patients out there whose lives could profoundly change if their own CT scans look something like these. The effects of Marfan syndrome affect me every day of my life, and I cannot begin to express how transformative a corrective therapy like this for Marfan syndrome would be. At the same time, I've also had to acknowledge that since I'm already an adult, any therapy that I play a role in developing likely won't benefit me personally. 72% of the 6,000 plus rare diseases are genetic, which means that 72% of rare diseases may, in the future, be candidates for genetic therapies to address the root cause of the disease, something that seemed truly unfathomable when I was growing up with Marfan syndrome. There are many other multisystemic disorders, even other connective tissue disorders like PXE, whose symptoms can improve with liver-directed gene editing. Maybe we don't have to wait for a new delivery technology after all. Maybe we just need to expand our ideas of which rare conditions we can tackle right now with the tools that we have. I can't promise whether or when therapies for connective tissue disorders will happen, but I can promise that I will continue to do what I can to give patients with connective tissue disorders and rare diseases in general a fighting chance at living longer, healthier lives. Thank you so much to the organizers at NIH and FDA who asked me to be a speaker at Rare Disease Day this year. I'm sorry that the event couldn't happen in person. Please feel free to reach out if you have any questions or comments. Thank you very much.